Mr. Chairman, ladies and gentlemen, thank you very much for this kind invitation. As we heard, the acute kidney injury and chronic kidney disease are not as separate entities, and there is a continuum of disease. Considering this new classification, acute kidney disease describes acute or subacute damage with the loss of kidney function, and this can have a duration between 7 and 90 days. We know that epidemiological association exists between acute kidney injury and the subsequent development and progression of chronic kidney disease. So there are some factors that are in favor of a progressive chronic kidney disease and also increase the cardiovascular mortality. So today I would like to show you some pathogenic mechanism that can be maybe targeted with the new drugs that maybe can modify the history of acute kidney injury. What the animal models told us in the research is that acute kidney injury produces a senescence-associated profibrotic secretory phenotype in the renal parenchyma, and the subsequent inflammatory milieu promotes the gradual accumulation of what? Of renal fibrosis, vascular rarefaction, and chronic kidney disease. However, in the definition of gradual accumulation, there is the concept that the chronic kidney disease and the fibrosis are present in the later time. Another important data from the literature is that the vascular rarefaction is present in 30 to 50% with a reduction peritubular capillary density and leads to hypoxia and tubular atrophy. This injured renal tubular cells, they acquire a senescent profibrotic phenotype with a cell cycle arrest, and two important cells that we will talk about today are pericyte and endothelial cells that are capable to transform to transdifferentiate in myofibroblast. So this is one of the last review summarized the data. But can we modulate this maladaptive cellular response, or is an irreversible destiny of the kidney? We have a model of ischemia reperfusion injury that is really frequent in kidney transplantation. And in this model, a very important role is played by complement system. In these very complicated slides, I only want to focus your attention on the fact that complement is not only important for cellular lysis by C5b2 but now there is much more data on the role of complement anaphylatoxin, especially C5a, and the C5a receptor are present in all the cells of the kidney and can induce a very potent pro-inflammatory response. So when we think about complement, we don't have to think to cellular lysis, but to a pro-inflammatory activity. And the complement system is really implicated in several kidney diseases, both in acute injury of glomeruli, vasculature, and tubules, but also in chronic inflammation. And now, several drugs are directed against the complement. We have this model in which we induce warm ischemia in pigs, and then we use a new complement inhibitor that has been tested in humans. Doesn't work in rats and mice because the magnitude of complement activation is much more powerful in pigs and humans, and that's been tested in humans. And then we start reperfusion, and what is interesting is that we perform kidney biopsy at early time point to see what happened. This is an images, and it is like to work with humans because these are pigs of 50 kilograms. And C1 inhibitor, you can see here, is an inhibitor of serine proteases and is capable to target both the classical pathway and the lectin pathway by blocking C1s and the MASP protein, giving a very good inhibition of these two pathways. And in our first paper, we demonstrate that these two pathways are really activated in ischemia reperfusion injury. You can see the deposit of C1q, the MBL, the C4d deposit, and the merge analysis. And this activation is especially active at endothelial cell level. When we analyze the tubular cells, you can see here that the tubulars undergo to apoptosis are protected by C1 inhibitors, so it's really effective in protecting the kidney by the ischemia reperfusion damage. Then we move to another cell that is really important, as I told you, are the endothelial cells. But endothelial cells are not really, when are activated, are not really undergoing to apoptosis or necrosis. Absolutely not. Endothelial cells do not die, but they have a maladaptive response. What is this maladaptive response? It's the endothelia to mesenchymal transition that was first described in chronic kidney disease. There were few data in acute kidney injury, and there is a transition from endothelial cells to an endothelial cell that express markers of fibroblast, and then in the end you have a capillary wall that is without the real endothelial cells and with an increase in myofibroblast. And this affects the microvascular density with the development of hypoxia. Depending on this data, we start to focus on endothelial to mesenchymal transition in acute setting. As you can see here, after 24 hours, endothelial cells, they already acquire FSP1, that is a marker of myofibroblast. What is also interesting to see is that we have a process of acute fibrosis. So the fibrosis is not a process that will start later, when chronic kidney disease started. But we can see sign of fibrosis with collagen deposits already at 24 hours. And when we block complement, we can block also the early development of renal fibrosis. And these data are quite interesting. And also because the C1 inhibitor has the capacity to bind damaged endothelial cells. So the drug is there where the endothelials can be activated by complement. We have shown also the occurrence of endothelial to mesenchymal transition in a model of sepsis-induced acute kidney injury in pigs. As you can see here, the acute fibrosis is present already after a few hours from LPS infusion, indicating, as you can see here, all the cells are the perivascular, peritubular capillaries. They acquire the phenotype of myofibroblast, and they start immediately to transform in a myofibroblast. As you can see here, this process of the endothelial to mesenchymal transition will start in renal vessels, but also in peritubular capillaries. So in summary, the fibrosis starts really early, already after a few hours from acute kidney injury. Then we move to the pericytes. Pericytes acquire important cells in renal parenchyma, and they are like an octopus, and they are around the capillary wall. And pericytes are considered another very important source of renal myofibroblast after acute kidney injury and in chronic kidney disease. We analyze in our model the eventual modulation of pericyte to myofibroblast transition. You can see here at basal level, pericytes, they have normal expression of PDGF receptor beta and very low expression of alpha-SMA. After 24 hours from damage, you can see a huge expression of alpha-SMA and a downregulation of PDGF receptor beta, indicating that pericytes are highly activated by acute kidney injury, and that C1 inhibitor can block the activation of renal pericytes. But more interesting, when we measure the area of peritubular capillaries at T0, you see here the normal area of peritubular capillaries. After 24 hours, the kidney looks like a liver hepatization process because all the capillaries are reduced in their volume. And this also depends in our model on complement because C1 inhibitor can prevent this closure of the capillary. And this difference is statistically significant and seems to be complement mediated. So in our hypothesis, when there is ischemia reperfusion and complement damage, we have the pericytes that will really constrict the peritubular capillaries, and we will have a negative effect on kidney function. But now what we can say from AKI to CKD, these are data on maladaptive repair. Is this an inevitable destiny? As you can see here, in this guy from Star Wars, he said to Luke Skywalker, it is inevitable, it is your destiny. This guy is a guy with accelerated senescence, with aging that is really advanced. And I think that in the aging is the response to the destiny of the kidney. And there are much, much more data indicating that after AKI, there is a senescent tubular epithelia with secretion of pro-fibrotic factors by G2M arrested cells. What is interesting about these senescent cells is that they can produce increased amount of IL-6, MCP1, so they start to have a pro-inflammatory phenotype, but also a pro-fibrotic phenotype. And this process is called inflammaging. It's not the physiological aging, but the cellular senescence can be accelerated by inflammaging. And also in this path, the complement is involved, because the complement is bad for aging, as in this paper and cell, the C1q is capable to induce the VNT signaling. This is really important for the senescence, for the cellular senescence. And when we focus on Cloto, we have to know that the kidney produces the much more powerful protein anti-aging, that is Cloto. And Cloto is produced by renal tubular epithelial cells. When we analyze in our model the Cloto at the basal level, you can see a very nice production by tubular cell of Cloto, but acute kidney injury can induce a downregulation of Cloto. So acute kidney injury will induce a senescent phenotype by abrogating this protein. Is this process also present in humans? The response is yes, because patients after transplantation that experience the telegraph function that is linked to ischemia reperfusion injury, they have significant loss of Cloto by tubular epithelial cells. But Cloto is also important because it is released into the circulation and has anti-aging activity also on the cardiovascular system and the central nervous system. So acute kidney injury can induce a persistent depletion of Cloto in the capacity of tubular epithelial cell to produce this important protein. When we start to analyze in vitro if this process is also occur, we see that C5a is really potent only with 3 hours to induce senescent cells in vitro. And this we can find also in vivo. So the complement activation tubular epithelial cells will induce senescent with loss of Cloto, that is an anti-aging protein. And the other characteristic of senescent cells can also be reproduced, since these cells are senescent, but they produce IL-6, MCP1, SCTGF. So they are really active in producing these molecules, even if the NOXA, if C5a has been removed. And this is in line with this interesting paper, where patients with sepsis associated with pneumonia were analyzed after hospital discharge. As you can see here, even after resolution of sepsis, the patient, they continue to produce pro-inflammatory cytokine. And this is a negative impact on cardiovascular mortality and also for neoplasia or for other syndromes. This means that after acute kidney injury, something is changed in our patient. And I think that epigenetic mechanisms are really, really important to explain the progression of acute kidney injury. For example, KIM1 and ICAM1 are epigenetically regulated by changes in DNA. So the acute kidney injury can give an insult that can modify our DNA in the capacity to produce or not produce some factors. And this is dependent on methylation that can be increased or reduced methylation that can activate some gene. As you can see here, some preliminary data from our lab, C5a can induce a significant alteration on DNA methylation in renal tubular epithelial cells. We will have 144 sites that will be hypomethylated, so will produce more cytokine or other factors, and other will be hypermethylation, so we'll start not more producing these factors. So what we can conclude, depending on this data, that there is a strong epidemiological association between AKI and the development or progression of chronic kidney disease. The link between AKI and CKD is supported by several experimental data, demonstrating that there is an activation of pro-fibrotic pathway, but we think that this pro-fibrotic pathway is already active in the acute phase. We can describe an acute renal fibrosis, and this acute renal fibrosis is characterized by endothelial to mesenchymal transition and pericyte to mesenchymal transition. This maladaptive repair after AKI shares common features with the kidney aging, and this is a state we think of accelerated kidney aging. And we think that finally the epigenetic mechanisms are the next target for future therapeutic intervention, try to modify the destiny of patients with acute kidney injury. Thank you for your attention.